Our last speaker of the symposium is Richard Lewis from Stanford University. Dr. Lewis is a pioneer in the study of ion channels and calcium signaling in the immune system, and this comes from his early work as a postdoc with Micah Halen at UC Irvine, where he studied biophysically the characterized calcium channel currents in T cells and thymocytes, which has really laid the foundation for this field of calcium signaling in many ways. He has continued to generate some of the most elegant and meaningful insights into the regulation of ORI channels and by the STIM1 molecules. And we thought, because of the importance of calcium signaling in immune cells and his expertise in crack channels, that he would be the perfect closing speaker for this workshop to talk about or to integrate lymphocyte function and signaling and ion channels. So Rich, thank you very much for doing this, and it's all yours.
All right, thanks, Stefan. Hello, everybody out there. Yeah, I feel like a voice in the wilderness here, but it's been a very long day. And in talking about calcium signaling and ion channels in immune cells, I'm going to try to connect some of the things that you've heard about today from immunologists and the physiologists, and hopefully provide some kind of a bridge between the two worlds. And I'm trying to particularly aim this at students in the audience, as they are potentially going to be the next generation of immunophysiologists. So I'll just start here. The first identification of ion channels in immune cells. Let me just move this around a little bit. Okay. Really started with the application of patch clamp techniques to record ionic currents in human T cells in 1984. And this was work from Mike Cahalan's lab and Carol Deutsch's lab. And this patch clamp technique made it possible to record and identify currents through ion channels in cells that were previously too small to study with sharp microelectrodes. And this applies to the cells in the immune system for sure. And their exciting results clearly showed the presence of voltage-gated potassium channels in human T cells. And they went on to show that these were essential for T cell mitogenesis. And at that point, the game was on. Since that time, we've become aware of in all innate and adaptive immune cells, many types of ion channels are expressed. And this diagram on the right just scratches the surface. I think there's surely more yet to be discovered. The ion channel universe is a complex one, and all types of ion selectivities, potassium, chloride, sodium, calcium, non-selective channels, many types of activation mechanisms and locations in the cell are all at work. Channels do a lot of things, a lot of housekeeping functions, as well as functions that are essential for the immune response. So I'm going to focus on channels that generate calcium signals. Calcium is widely known as one of the most promiscuous second messengers. Every cell depends on calcium for its physiological behaviors, and immune cells are no exception to this. Mechanisms and consequences of calcium signaling have probably been most extensively studied in T lymphocytes, and so that's what I'm going to focus on, while I try to illustrate basic mechanisms and the overall logic of calcium signaling networks that might give a starting point to approach signaling in other types of immune cells. So a rise in cytoplasmic calcium is one of the early events that occurs within seconds of when the T cell recognizes antigen on the surface of an antigen-presenting cell. This is an essential signal for T cell activation. This movie from the Cahalan lab from years ago now shows a T hybridoma cell in vitro loaded with FIRA2 crawling around, and when it happens to contact the B cell target, its calcium quickly increases. Now, calcium signals are involved in some of the events you can see, like the stopping, forming a synapse with the antigen-presenting cell, as well as some things that you can't see, like activation of gene expression programs, later proliferation and secretion of cytokines or cytotoxic factors, which I'm going to get to later. But first, I'd like to talk about the overall organization of the calcium signaling machinery and how ion channels and pumps act to generate and shape these signals. Calcium signals are generated by basically the imbalance of calcium sources and sinks. There are several sources. There's the extracellular space. There are internal organelles like the endoplasmic reticulum, mitochondria, endosomes, and lysosomes. And as you heard from Lily Jan, the calcium flux through channels in these membranes is governed by electrical and chemical driving force. ATP-driven pumps in the plasma membrane extrude calcium and set up concentration gradients. So they load the ER and endolysosomes to about 0.5 millimolar internal level of calcium, while reducing the cytoplasmic level to about 100 nanomolar, thus creating a 5 to 10,000-fold concentration gradient for calcium for flow across these membranes and into the cytosol. In addition, there's a minus 60 millivolt membrane potential across the plasma membrane set up by potassium channels and a potassium gradient that's generated by, again, another ATP-dependent pump, in this case the sodium-potassium pump. And this is energetically, minus 60 millivolts or so is energetically equivalent to about 100-fold concentration gradient for calcium. So it supplies an additional boost, a significant boost to calcium entering the cell. Mitochondria have even a higher electrical gradient driven by the electron transport chain, about a minus 180 millivolts, that drives robust entry of calcium through the mitochondrial uniporter, MCU. And this allows mitochondria to accumulate calcium to high levels and, as we'll show later, affect calcium-dependent processes that are located nearby. So how does this process begin with antigen recognition? Oh, yeah, well, there's the electrical driving force and the concentrations. Okay. So how is this process initiated? Well, as you heard from Leslie, there's a series of biochemical events that occur following the binding of antigen to the T cell receptor. And in the end, this activates phospholipase C to produce IP3, which releases calcium through IP3 receptors located in the ER. And Steve showed a beautiful example of the structure of this huge protein earlier on. It turns out that other channels may be involved as well. So there's an early generation of the messenger NAADP, which may actually first initiate calcium release from the ER through ranitidine receptor type 1. And it's thought that the release of calcium through this receptor might synergize with IP3 to help open the IP3 receptor. Now, this NAADP pathway is less well characterized than the IP3 pathway. For example, the enzyme that creates NAADP has not yet been reported, but recent work has uncovered a protein H1NL or GPT2 in the ER that appears to bind NAADP and couple it to activation of the ranitidine receptor. But regardless of the precise details of this mechanism, the release event, as shown here, produces only a transient calcium increase in the cell. These are Jurkat cells stimulated with an anti-CD3 antibody in the absence of calcium. And you can see at most one or at most a few transients, small transients of calcium that are produced by this stimulation in the absence of extracellular calcium. In order to sustain calcium for the minutes-long time that's needed to activate the cell, calcium influx is needed. And it's worth noting that the sustained calcium in T cells can take several forms. At high stimulation levels, as seen here, such as a high concentration of antibodies to cross-link the T cell receptor in vitro, or high avidity interactions in vivo, responses often show a peak and a sustained plateau. While low stimulus strength in vitro will elicit baseline fluctuations or oscillations. Now, the advent of two-photon microscopy and genetically encoded ratiometric calcium indicators has now made it possible to record the similar signals from cells in physiological settings as they undergo immune responses. This example here is from ova-specific T cells in a lymph node in vivo after the animal has been challenged with injection of the ova peptide. What you can see in the red traces are the calcium signals, which are sustained for quite long periods, tens of minutes, with some fluctuations that seem to be correlated with high calcium, correlate high calcium with low motility and vice versa, suggestive of a stop signal that may be calcium dependent in these cells. Well, where does the TCR mediated calcium influx come from? Well, you've heard this several times already, and I feel like it's almost a broken record at this point, but I'll continue. They come through store-operated calcium channels. These are channels, a class of channels that are present in just about every cell type in the body, and they're activated by depletion of calcium from the lumen of the ER that follows IP3-induced calcium release. They can be activated artificially by inhibiting the calcium pumps, the circa pumps that load the ER with calcium by compounds such as thapsigargan. And down here, what I'm showing is that in a T cell from a healthy person, application of thapsigargan in the absence of calcium releases the load of calcium from the ER, creating this small signal. And then once that calcium has been released and the ER is depleted, the readdition of calcium unmasks a great influx from the outside, which is through these open store-operated channels, the crack channels. The same thing happens in healthy T cells if the cells are instead stimulated by TCR cross-linking, in this case by anti-CD3 biotin and then cross-linking with streptavidin. You can see in the absence of calcium, the IP3-induced release, and then when calcium is re-added, a large influx of calcium from outside. Now in SCID patients having a defect in the crack channel, non-functional crack channel, neither thapsigargan nor T-cell receptor stimulation evokes a calcium rise. And this is an important result because it shows the high degree to which these TCR-mediated signaling triggers store-operated channels as opposed to many other potential calcium influx pathways. This is probably something we'll discuss later on. Now, looking at the current directly through the crack channels by whole cell patch clamp recording has produced a detailed biophysical fingerprint of the channel properties. Now, in the interests of time, I'd only like to mention here a couple of distinctive things about this channel that are particularly relevant to its signaling functions. You've heard some of this from Steve Long earlier today. First, this is an extremely calcium selective channel, about 1,000 to 1 selective for calcium flux over sodium. And this is indicated in one sense by this inwardly rectifying current voltage relation. So the point is that this is essentially a pure calcium current. There's a negligible amounts of sodium that are going to enter along with calcium. And this minimizes the depolarization of the cell membrane that would occur when the channel opens. The second characteristic is that it has an extremely small conductance. And Steve talked about this a little bit earlier, and perhaps a structural basis for this behavior, or this characteristic. The conductance is about 30 femtosiemens, which corresponds under physiological conditions of something like 10,000 calcium ions passing through, entering the cell per second. This is about 100-fold less than the flux of calcium that one would expect through, say, a voltage-gated calcium channel. So this is really a dribble of calcium, which makes it suitable perhaps for long-term calcium signaling without overload of the cell, as well as local signaling to achieve some specificity in its downstream targets. This is something I'll get back to later. And finally, I'd like to point out that the channel is not voltage dependent. It is really dependent on the ER calcium level, and this has been measured. by loading calcium indicators into the ER and measuring, depleting the ER by various amounts, measuring the crack current. And you can see that the ER really has to lose about half of its load of calcium before this channel really starts to turn on significantly. So the overall way in which calcium depletion in the ER activates the channel is now relatively well understood, but there's still some mechanistic details to work out. Luminal calcium is sensed by stim proteins in the ER membrane through binding to a luminal EF hand. There are two paralogs of stim 1 and stim 2, that differ in their calcium affinity. Stim 1 is the main isoform involved in activating crack channels, particularly in human T cells. Stim 2 has a lower calcium affinity, so it stays active, partially active, even at resting levels of calcium in the ER, and is thought to respond more to small fluctuations of calcium around the mean. The CAD domain, or crack activation domain, of stim 1 binds to the Array proteins in the plasma membrane that make up the crack channel and causes them to open. This CAD domain is sequestered in the resting state by a coiled coil 1 domain, or CC1 domain. But after store depletion, stim changes its conformation, releasing CAD and allowing it to interact with the crack channel. Stim1 and Array1 are the main isoforms found in human T cells, but there are others that are important in other cells and under different conditions, and I'll talk about this later. So the way in which stim and Array interact is really quite unusual for an ion channel activation mechanism, and it's as both proteins are highly mobile in the resting state. So in resting cells with full ER calcium stores, stim1 dimers are in an inactive conformation, diffusing freely throughout the ER. Array1 hexamers, comprising the crack channels, are also diffusing in the plasma membrane. And ER depletion evokes a conformational change in stim1 that exposes a polybasic domain that allows it to interact with PIP2 in the plasma membrane. And this concentrates stim1 at these ER plasma membrane contacts, while the CAD domain is now revealed and allowed to interact with Array, which traps the channel at these same locations and opens it to stimulate or allow calcium entry. Now, this movie here shows this process at the light microscope level. And what you can see is that it's the footprint of an HEK cell expressing cherry-labeled stim1 and GFP-labeled Array. I'll show that once again. And when an inhibitor of the SERCA pump is added to the cells, the ER becomes depleted, and stim and Array co-localize in these puncta. So there goes on the inhibitor, and within minutes, both proteins co-localize in these bright puncta. And by looking at the ultrastructural level at the distribution of HRP-labeled stim1 in Jurkat cells, it's been shown that these puncta actually correspond to ER plasma membrane junctions. And you've heard about these again today. These are specializations of the ER where it comes to within 10 to 20 nanometers of the plasma membrane. And this distance of 10 to 20 nanometers is critical because it's the only place in the cell that is close enough for stim1 to make physical contact with Array1 in the plasma membrane. These junctions in resting Jurkat cells cover only about 5% of the cell surface. Now, this mechanism of activation is unique for an ion channel, where the different parts of the channel complex are located in different membrane compartments and only assemble after arrival of the stimulus. But there are two key aspects of this arcane mechanism that I'd like to come back to, and that is that it's dynamic and flexible. Junctions can form, they can move around inside the cell, they can disassemble, and when they do this, they change the site of calcium entry, which actually occurs as T cells form synapses on antigen-presenting cells. And Also, these junctions are important in that they create a restricted space in which calcium enters the cell before it can diffuse out to enter the bulk cytoplasm. And this may be important for achieving specificity and efficiency in activating some downstream effectors, such as the transcription factor NFAT. So I'm not going to talk too much more about the mechanism. I'd just like to note that the linchpin in this entire process, should go back, the linchpin in this entire process of store-operated calcium entry is the STIM protein and its transition between the quiescent and the active conformation. Once STIM becomes activated, everything else occurs autonomously by a diffusion trap mechanism. So the full structures of STIM1 in quiescent and active states are not available at this point, as the protein seems to be very flexible. But a variety of evidence, including some recent single molecule FRET experiments that we've done, measuring the conformation or assessing the conformation of the inactive state, suggests that when calcium is bound, this CC1 domain is arranged around the CAD to keep it pointed towards the ER membrane. And the release of calcium from the luminal domains allows the EF-SAM domains to come close together and release the CC1 domains from CAD. While this exposes the CAD, while coiled coil forms in the transmembrane domains and in the proximal CC1 domain. And this extends the CAD towards the plasma membrane to interact with Ri. By introducing pairs of cysteines at various positions within the CC1, we found that these cysteines can form disulfides in the active state of the protein, suggesting that there's a close pairing between these various CC1 helices along their entire length in the active state. So much remains to be learned about the precise structures of STIM1. There's a crystal structure of the CAD domain and of the EF-SAM domain in isolation, but nothing else. And it's also not known how the interconversion of these two states is controlled and what the intermediates are, as well as the details of how the CAD binds to Ri and how this binding leads to channel opening. So there are many, many important questions about structure and function that remain to be solved with this important protein. So I'd like to turn now to what crack channels actually do in T cells. A lot has been learned from loss of function mutations in human patients. In human T cells, Ri1 and STIM1 are the predominant isoforms, and so that knocking out either one or getting loss of function in either one is sufficient to completely abrogate store-operated calcium entry. It turns out that in mice, this is not the case, that Ri2 is in many T cells in mice the predominant isoform. And STIM2 also makes a larger contribution. So loss of function in either STIM1 alone or Ri1 alone doesn't eliminate store-operated calcium entry. And in fact, one needs to do a double knockout of Ri1 and 2 or STIM1 and 2 in order to get a complete loss of calcium entry. Now, a lot's been learned about crack channel functions from these double STIM knockouts in mice. And it shows defects in the development, differentiation, and effector function of T cells. So the development of some T cell subsets in the thymus is independent of store-operated calcium entry. For example, there's normal alpha-beta T cell, CD4 and CD8 T cell development in the absence of store-operated calcium entry. But interestingly, the regulatory T cell arm is highly dependent on store-operated entry for development in the thymus. And furthermore, the differentiation of these thymus-derived Treg cells into effector regulatory cells in the follicle where they stimulate, where they regulate the germinal center response, as well as tissue-resident Tregs. This all requires store-operated calcium entry. And the lack of Treg cells, as well as their reduced ability to produce immunosuppressive cytokines like IL-10 and IL-35 may explain the autoantibodies that were seen in the human patients with the crack channel loss of function. The defective function of TNF-alpha and interferon gamma by store-operated entry deficient Th1 and CD8 positive cells probably contributes to defects in antiviral and anti-tumor immunity that were observed in the human patients. Now, an effective antiviral response requires store-operated entry in both CD4 and CD8 positive cell subsets. In the CD8 positive cells, SOCE controls the release of cytotoxic granules containing granzyme B and perforin, as well as the expression of Fas ligand and inflammatory cytokines like TNF-alpha and interferon gamma. Strong suppression of store-operated calcium entry also interferes with the generation of effector and memory CD8 positive cells. And all of this conspires to make acute viral infections chronic, which is another characteristic of human patients with the crack channelopathy. Now, a surprising finding was that store-operated calcium entry and calcineurin also direct the metabolic reprogramming of T cells as they prepare for entering the cell cycle. So normally cells will transition from oxidative phosphorylation to glycolysis after antigenic stimulation. This STIM1, STIM2 double knockout cell failed to express multiple types of glucose receptors, the GLUT1 and GLUT3 family, the glucose transporters, as well as glycolytic enzymes, which impaired glucose uptake, glycolytic metabolism, and entry into the cell cycle. And this was traced to defects in NFAT activation and the PI3 kinase, AKT kinase mTOR nutrient sensing pathway. And this could explain why T cells from crack deficient patients failed to proliferate in response to mitogens in vitro, and why calcineurin inhibitors like FK506 and cyclosporin A inhibit proliferation of T cells. So you probably noticed by now that many of the functions of store-operated calcium entry that have been gleaned from the STIM1, STIM2 knockouts mice involve the activation of NFAT by calcium activation of calcineurin, which dephosphorylates NFAT and exposes a nuclear localization sequence that allows it to enter the nucleus. Calcium also activates other parallel calcium effector pathways involving CAM kinase, ERK, and NF-kappa B. Now, while it's known that NFAT translocation into the nucleus is controlled by calcineurin, there are several studies that show that with physiological rises in calcium, the bulk of this reaction doesn't occur within the bulk cytosol, but rather within local nanodomains, very close to crack channels, within the cleft of the ER plasma membrane junction. So one indication of local activation is from comparing NFAT translocation into the nucleus in response to calcium oscillations evoked in the presence or absence of calcium influx. So in many cells, stimulation of phospholipase C-linked receptors will evoke calcium oscillations. This is caused by repetitive release of calcium through the IP3 receptor in the ER membrane. And because plasma membrane pumps pump out a substantial fraction of the calcium that exits through each spike, during each spike, crack channels need to open in order to resupply the calcium that's been lost to the cell. Otherwise, the oscillations die out over time as the ER becomes depleted of its load of calcium. So interestingly, applying high concentrations, millimolar levels of lanthanides to the bathing solution will not only block crack channels, but also inhibit the plasma membrane calcium ATPase. So now it's a closed system in which all of the calcium from the oscillation can then be recovered from the cytosol and pumped back into the ER by the Circa pumps. Essentially, the same oscillations occur, but without any influx from outside. This experiment shows that oscillations that are evoked in HEK cells by LTC4 with store-operated calcium entry active, in the presence of calcium, causes NFAT translocation. But nearly the same oscillations evoked when calcium entry is blocked do not suffice here. So the same global calcium signal doesn't send NFAT to the nucleus. It needs local calcium coming through the crack channel. In other experiments, it's been shown that introducing a slow binding calcium buffer like EGTA into the cell can inhibit the bulk calcium rise, but doesn't affect the response of NFAT to calcium entry, again consistent with a local effect on NFAT. And recent work from the Perec lab has shown that the scaffold ACAP79 is an essential player in this process. ACAP79 in RBL cells binds a fraction of the NFAT and calcineurin pools, and store depletion causes it to associate with ARI1 by way of a short sequence located in the N-terminus of the channel. And interestingly, this sequence is only present in ARI1, but not ARI2 or ARI3. And accordingly, cells expressing only ARI2 or ARI3 fail to effectively translocate NFAT to the nucleus in response to thapsigargan. So this is an interesting result, as it suggests that ARI1 may be more capable of activating NFAT-dependent pathways than the other isoforms, which could be significant in systems like mice where the expression of ARI isoforms changes as the cells differentiate. But another aspect related to the specificity of signaling at the ER plasma membrane junctions is that only a few endogenous crack channels are thought to be active at individual junctions, and each conducts only a dribble of calcium into the cleft, further adding to the specificity of activating processes within the local nanodomain. So now I'd like to describe a somewhat surprising function. Let's go on to this. A surprising function of crack channels in supporting the organization and function of actin at the immune synapse. And I'm going to have to go into play mode here, and I just hope nothing happens. So after contact with the antigen-presenting cell, the T cell spreads over the target cell surface and reorients the ER and mitochondria towards the contact site. At the synapse, actin reorganizes into a branched lamellipod at the periphery of the synapse, with circular arcs of actin containing myosin in the lamella. Actin treadmilling and arc contraction continually sweep TCR microclusters, shown here, from the periphery towards the center of the synapse, the so-called CSMAC, where they're endocytosed and then recycled to the periphery. The movement of actin appears to be necessary to promote T cell receptor activity and the enzymatic activity of these microclusters. This movement can be visualized by TURF in an artificial system of plating Jurkat cells on an anti-CD3 coated cover slip. And you can see the organization and movement of actin on the left, myosin in the middle. These are, this treadmilling and concentric constriction of the actin-myosin arcs towards the center of the synapse. And it turns out that this is what's driving the TCR microclusters towards the center. And the surprise is that the crack channels are essential to keep all this going. So in the top right, if I can play this movie, what you see is actin in a Jurkat cell. We're looking at the footprint again. And it's treadmilling, and we'll just remove acutely the calcium from the bath, and you can see what happens there. You lose the organization and the dynamics. When we apply calcium back, it's rapidly restored. So it turns out that crack channels, that calcium flux through the crack channels, is necessary to maintain this organization and dynamic movement of actin at the synapse. Now, this movement of actin appears to confine the ER and stim and ARI to the center of the synapse, such that newly formed stim-array clusters at the periphery of the synapse can be seen to move inward towards the center. Now, the particular function of these highly localized channels is not yet known, but it coincides with the location of exocytosis and endocytosis at the junction with the antigen-presenting cell. So it's tempting to think that they're positioned in this way to optimize those activities. The details of the mechanism haven't been worked out, but calcium increases. Let me just show you this first. This shows that in the absence of calcium, as it will recycle here in a second. Come on. Here, in the absence of calcium, ER tubules extend all the way to the periphery, but once calcium is present, the advancing wall of actin pulls the ER tubules back towards the center and concentrates the ER and ER plasma membrane junctions and stim and array to the center of the synapse. Now, we haven't worked out the details of the mechanism yet, but we found that calcium increases actin depolymerization, which provides a source of free monomeric actin, while suppressing polymerization in the lamella and confining polymerization to the outer edge of the synapse. And this highlights a self-organization mechanism by which calcium influx through the channels serves to concentrate the channels at the center of the synapse. Now, there are a variety of other channels and pumps that are essential for promoting store-operated or modulating store-operated calcium entry. And I just want to talk briefly about some of those now. Several channels can affect calcium entry by altering the driving force. KV 1.3 and KCA 3.1 channels enhance the driving force for entry by outward flow of potassium, while the TRPM4 channels, relatively non-selective, conduct inward flux of sodium and depolarize the cell to decrease the driving force for calcium. And this can be appreciated from the current voltage relations of each of these channels. Any inward movement of calcium through the crack channel would tend to depolarize the cells, activating potassium efflux here and also here, and this outward current will tend to repolarize the cell, while the calcium elevation activating TRPM4 activates this non-selective current, which at resting potentials of the T cell around here is going to conduct an inward current which tends to depolarize the cell. Now, the effects of the roles of the K channels on calcium signaling and T cell function have been nicely dissected through the use of highly specific blockers, such as the CNM toxin SHK for the KV 1.3 and TRAM34, a blocker of the KCA channel. And it turns out that these two potassium channels play overlapping roles to some extent, so that knockout or block of KB 1.3 doesn't really affect acute immune responses in rats. But upon repeated stimulation of cells in vivo, akin to booster shots for a vaccination or constant stimulation of autoreactive cells in autoimmune disease, the cells become dependent on KV 1.3. And this is because in patients, for example, with rheumatoid arthritis, the activation of cells in the synovial fluid causes an upregulation of KV 1.3 channels from about 300 to 1500 channels per cell, but not KCA 3.1. Whereas the activation of central memory T cells that are not involved in disease upregulate KCA 3.1 but not KV 1.3. So the interesting finding is that a KV 1.3 blocker selectively suppressed this effector memory cell function, both in terms of suppressing calcium influx, suppressing proliferation of the cells, and suppressing cytokine production by these cells. So by preferentially suppressing autoreactive effector memory T cells, these specific KV 1.3 blockers may have a therapeutic potential for treating autoimmune diseases in which effector memory T cells play a role. Now, TRPM4 is a non-selective calcium-activated cation channel. As I said before, the rising calcium opens it, creating an inward current that depolarizes the cell. And its effects on calcium signaling and T cell responses have been studied by expressing a dominant negative form of TRPM4 and that inhibits the endogenous current. In controlled Jurkat cells, treatment with phytohemagglutinin, which stimulates the T cell receptor, evokes typically a series of calcium oscillations, characteristic of a moderate response. But when the channels are suppressed by the dominant negative construct, the same treatment evokes a much larger and more sustained calcium rise. And this effect was accompanied by an increase in the production of IL-2 by these cells, suggesting that depolarization by TRPM4 normally acts to reduce calcium signaling and control the magnitude of downstream activation events. Now, mitochondria also play a complex role in modulating calcium entry, apparently through several mechanisms. As I mentioned before, they move to the synapse along with the ER and the microtubule organizing center. And the first thing they do is they suppress a slow calcium-dependent inactivation of the crack channel that turns off the channel by taking up calcium through the mitochondrial uniporter and reducing buildup of calcium around the channel. In addition, the calcium that they take up activates dehydrogenases within the lumen, within the matrix, to boost ATP production, which is a way of matching ATP generation to metabolic need. Some of this increased ATP is thought to leave the cell through pannexin 1 channels, also located at the synapse, that conduct calcium into, and that acts locally to locally activate purinergic receptor channels like P2R, X1, X4, or X7, that then can conduct calcium into the cell and boost the cytoplasmic calcium signal. And finally, the mitochondria can also enhance calcium signals by reducing efflux through the plasma membrane pump. Specifically, by taking up calcium, they reduce modulation of the primary pump PMCA4B. Now, it's known that PMCA4B in resting cells is largely autoinhibited, but that this inhibition is removed over seconds following a calcium rise by the calmodulin-mediated displacement of this autoinhibitory domain. The uptake of calcium by mitochondria reduces the uptake of the upregulation of the pump, shown here at the immune synapse, enhancing more sustained calcium signals at the synapse. So I think I'm running up on the time limit here. I should say that this modulation process can be observed directly in a perforated patch configuration where the cytoplasm is relatively unscathed and electrical access is provided through small antibiotic pore-forming peptides that incorporate into the patch under the pipette. And in this kind of situation, one can use voltage to create a constant inward current through the crack channel, through open crack channels, and you can see that the internal calcium, the cytoplasmic calcium increases, but then decreases as the pump becomes modulated and increases its activity. And I didn't point this out before, but this just shows the localization over time of mitochondria towards the synapse in a T cell. Okay, so I think I'm going to stop there. I think it's clear that the control of calcium levels in T cells, in fact in all cells, is the result of a complex interplay of sources and sinks, many of which operate within prescribed local microdomains and involve protein-protein interactions, diffusible messengers, and driving force effects on calcium flux. I haven't discussed by any means all of the calcium pathways thought to operate in T cells, some of which are controversial and others not very well understood at this point, but I'm happy to take questions. And thanks for listening.
Thank you very much, Rich, for the very clear lecture. There are a couple of questions. Maybe I can start with the last one because it's also one of your last slides. What happens to the calcium signal and in MCU knockout mice? Do you know that? Or especially, I guess, crack channel function and SOCE.
Right. Well, so the if one blocks the uptake of calcium by mitochondria, say by knocking out MCU, it does have an effect on the slow calcium-dependent inactivation. So in the absence of the calcium uptake, the channels will undergo the slow inactivation process.
Okay.
I don't know, the immunological, if you're getting at the immunological manifestations of that, I don't think those have been looked at.
Well, that was a question from the audience. I mean, I had the same question,
Yeah.
but that was a question from the audience. Another question is how can mitochondria slow CDI when they're too big to fit into ER plasma membrane junctions?
Well, there are a couple of types of inactivation. There's a fast and a slow process, and they're very distinct. The fast process is very local to the channel and probably involves calcium binding within a few nanometers of the pore. And mitochondria do not affect that. In fact, mitochondria get to maybe within 100 nanometers or so of the channels, but they're excluded from the ER plasma membrane junction by their size. The slow calcium-dependent inactivation, which is what I was talking about, is affected by mitochondria and is also affected by, can be prevented by slow calcium buffers like EGTA, which can only intercept the calcium after it's traveled, maybe 100 or 200 nanometers from the channel. So that's consistent with some sort of mechanism where calcium is acting at a site relatively distant from the channel to affect its activity, and where mitochondria can affect that. But so far, there's no real convincing mechanism that would explain that sort of spatial organization to the inactivation process.
One question that was asked in the chat was: is there mechanosensitive calcium channels such as piezo that plays a role in T cells?
Yeah. Well, people are just starting to look at that. And there's definitely evidence from Mike Cahalan's lab and others that stimulation of piezo channels will evoke calcium rises in T cells. And, one can easily imagine situations, as T cells are migrating through lymphoid tissue where they're subjected to all sorts of random tugs and pulls on the membrane. And in fact, there have been observations of ongoing small calcium responses, flickers and things like this as T cells are migrating through the lymph nodes. And some very nice work from the Cahalan lab suggesting that these ongoing small calcium rises are necessary to maximize the efficiency of T cells searching for antigen. I don't know if those signals are actually attributable to piezo-1, but it wouldn't surprise me if these kinds of stretch activated channels are going to play a role there.
There is one question about TRPM7, which is obviously a very important channel in T cell function. It's, as you know, it's a non-selective channel that can conduct magnesium, but also calcium. And Ingo Lang is asking, considering indications that TRPM7's kinase function regulates myosin 2A and also ORI-mediated currents, do you think there's a direct interaction between TRPM7 and ORI channels?
Well, I have to claim ignorance on that. I haven't seen any evidence for that. It's not out of the question. And the TRPM7 chanzyme, with the channel domain and enzyme domain, is really a peculiar kind of protein. And there's still, as far as I know, People are starting to parse out the different functions of those two activities, but it's a complex issue.
There's two questions, I guess, related to your the story about the rearrangement of all the microdomains in the immune synapse formation. One is about neutrophils. I'm not sure if you want to answer that. It says, interestingly, the proximity of ORI1 to focal adhesions in neutrophils also regulates cytoskeletal activity, but the rate of calcium influx is much faster than in T cells. What might control rates of flux that are also cooperative with ER stores?
I didn't hear the last part.
The question part of that comment is, what might control rates of flux that are also cooperative with ER stores?
It's also I'm sorry.
Yeah, so I'm not quite sure I understand the question correctly.
Okay.
But the statement part of the question is that the calcium flux rates in neutrophils are faster than those in T cells at focal adhesions. And then the question is, what might control rates of flux that are also cooperative with ER stores?
Cooperative with what?
ER, endoplasmatic.
With ER.
Mm-hmm.
Well, if I understand the question correctly, I mean, it's certainly possible that any kind of flux across the plasma membrane, whether it's integrin mediated or what, if it's located close enough to the ER, could interact with ranitidine receptors and even possibly IP3 receptors to cause calcium-induced calcium release. And so there's a potential synergy there.
Mm-hmm. Another question, and I have to read this to you, and this is a complex question that by Lily John. It says polarized release of T-cell receptor-enriched microvesicles at the immune synapse happens at the center. A possible relation with actin constriction to move ER to the center of immune synapse.
Yeah, I mean, we do think that based on observations of the ER extension at the synapse, which is basically being pulled out continuously by microtubules that are extending towards the periphery, we can see individual tubules that encounter this actin fence as it proceeds towards the center, and it will turn the tubule and cause it to actually bend and then retract, come back to the center. So I think, again, there are a lot of mechanistic details to work out in terms of the cell biology. To the eye, it seems like this advancing wall of actin is sufficient to pull the ER back into the center. But there are other possibilities as well. So,
thank you, Rich, for this trying to pull all this together, calcium signaling in T cells and immune cells. I appreciate it.